All right, class. Hello. This is second lecture on the lymphatic immune system. I'm going to share my screen and we're going to get started here. All right, last time we left off, we had done, it's working, other screen here, and so it's mirroring. Okay. Let me get my annotation little spotlight thing. There we go. Cool. We had left off going through the three different types of lymphocytes. We talked about B cells, we talked about T cells, and we talked about natural killer cells. Okay, so these are the three types of lymphocytes, and they work to provide adaptive immunity. And so last time in class, we did a brief overview of the immune system. We talked about the innate system, innate immunity versus the adaptive immunity. And we're going to talk about how the innate immune system communicates to the adaptive and prepares it. Before we do that, we're going to do a review of the some of the anatomy of the lymphatic system. So the lymphatic immune system incorporates the aspects of the lymphatic system, which deals with lymph, and then the lymphoid tissue, which produces lymphocytes. Okay, so lymph processing, lymph is a fluid. We need to move fluid. So that's lymphatics. Lymphoid, okay, that has to do with cells. So they both have lymph in them, the phrase or the word. So don't confuse lymph with lymphoid or lymphoid cells, okay, lymphocytes. Okay, one's a fluid thing, one's a cell thing. The fluid was named for the cells. And so it's a little bit hard to detangle those. All right, anyways, lymphoid stem cells. We talked about how all white blood cells are born. They are produced in red bone marrow, okay? B cells are produced there and they develop in the bone marrow, also natural killer cells. So in some sense, B cells and natural killer cells are more like each other than natural killer cells are like to T cells, okay? B cells and natural killer cells, they're both are developed in the bone marrow, okay? Group 2 are the T cells, okay? Once the T cells are produced in the bone marrow, they travel to the thymus, and that's where they develop as T cells. And they develop within what's called the blood-thymus barrier, because that is a very much a reinforced barrier between the blood and the thymus tissue, so that these premature T cells cannot escape and begin circulating in the body and potentially causing problems like an autoimmune reaction. Right, so this is just a graph showing the natural killer cells and B cells coming from a common lineage, and then you have T cells. So the peripheral tissues have the natural killer cells always are moving around, checking on our natural cells. Okay, so imagine natural killer cells are just always like roving policemen, as it were, and they were just forming cell-to-cell interactions and checking on natural cells to see if they are displaying the right type of cell surface marker. Okay, and if that cell surface marker is appropriate, then the natural killer cell just keeps on going. But if the cell surface marker of our natural cells is not appropriate, then the natural killer cell will get involved and potentially initiate a kill sequence on the abnormal cell. Okay, so this is a kind of an iffy analogy, but it's like if you have your documentation, like if you're a US citizen, you should have a social security number, a driver's license, some type of formal identification that proves that you are a citizen. Birth certificate, obviously, the birth certificate is a proof of citizenship because it shows that you were born here and you have certain rights that go with that. So if someone goes around and was checking like, hey, can I see your documentation? Yep, here's my documents. I'm supposed to be here. That's the way cells kind of act to natural killer cells, because they're always kind of showing, here's my documentation, I'm supposed to be here. And then the natural killer cell checks it out. Yep, that's appropriate. And then they keep moving. But if a cell displays documents, okay, these cell surface markers that are inappropriate, then the natural killer cell is like, wait a second, that doesn't look right. You don't have the right type of cell surface marker, and I'm going to begin to get suspicious, and there's going to be some other mechanisms for these cells to confirm whether this is a foreign, abnormal, or harmful cell. We also talk about antibody-mediated immunity. That is when the naive B cells become activated into plasma cells, they secrete antibodies, okay? And the antibody is a secreted protein that floats around in the blood and in the tissues, sometimes in the mucus, and other mucous membranes can be filled with antibodies to provide the protection that antibodies give us. And then cell-mediated immunity, okay? This is our cytotoxic T cells, okay? Remember the CD8 positive cells? CD8 has these two circles that are touching, like the numeral 8. Cell-to-cell combat, that's a cell-mediated immunity, okay? Cytotoxic T cells. All right, so these B cells and T cells, they can migrate throughout the body and in the tissues. So they're migratory. The other important thing about lymphocytes that you need to know is that they retain the ability to divide, okay? They can undergo cell division, and they do something called clonal expansion, okay? Clonal expansion means that once that B cell or T cell gets activated, it will begin to receive a signal to rapidly divide, make more of itself. The B cells will make more of itself so it can make more antibodies. The T cells will make more of itself so that it can attack a very specific pathogen. So other white blood cells do not have this ability, okay? So neutrophils and eosinophils and monocytes do not have the ability to undergo cell division, but lymphocytes do, okay? So that's an important cellular difference there. Hold on a second. Righty. So let's review a little bit more about the anatomy of some of these tissues and organs that are associated with the lymphoid and lymphatic systems around the body. We're going to talk about lymphoid tissues. These are connective tissues dominated by lymphocytes. That is, it's a cluster of lymphocytes in a particular region of the body, but not its own organ. Okay, lymphoid nodules and tissues are kind of synonymous in this respect. Okay, lymphoid nodules will have what's called a germinal center, and that allows for B cells to get activated and then go into what's called this clonal expansion. That is, they get activated and they start making more and more copies of themselves. Okay, so first area where we talk about this is the tonsils. Okay, the tonsils are five lymphoid tissues that are surrounding the pharynx, the mouth, the nose. They're at the back of the oral cavity and the nasal cavity in the pharynx. There's 5 locations. You have 2 lingual tonsils, you have 2 palatine tonsils, and you have 1 pharyngeal tonsil. They're all located in the back of these cavities in the pharynx. What is a tonsil? It's a collection of lymphoid tissues which contain B cells and T cells. Okay, if you think about the mouth and the nose, what's commonly going in and out? Well, food and drink and air. Okay, so we're continually sampling our environment through what we eat and drink and the air that we breathe. And so if there's stuff coming into our body that is harmful, pathogens, microbes, viruses, then these lymphoid tissues, the tonsils, can continuously sample the air and the food and the drink, and then bring in and kind of test those samples to see, is anything entering the body that could be potentially harmful? And then if the tonsils get activated, like, oh, there's something dangerous here, then they can respond by doing antigen presentation and cycling up a stronger immune response for the adaptive immune system. Okay, so tonsils are tissue, they're lymphoid tissues that are in the neck, the throat, and the mouth. This is what it would look like on the surface. You can have the intestinal lumen and the mucosa here. So that's a mucus layer. Just below the surface, you can have these collections of lymphocytes. Okay, this blue blob here represents a a lymphatic tissue, just a big collection of B cells and T cells. Okay. Here's what that would look like in a histological section. You can see if you zoomed in here on this, you would see a bunch of tiny little lymphocytes just all clustered together and awaiting any signal from a dendritic cell or a macrophage. So where are the other of these lymphoid tissues? You have lymph nodes, which we're going to talk about. We're going to talk about the spleen, the tonsils we just talked about, and then along the digestive, urinary, and reproductive glands, we have what are called MALTs, M-A-L-T, which is the mucosa-associated lymphoid tissues. So let's go through some of these. Okay, here's those MALTs. Those are lining the GI tract, the urinary tract, the reproductive tract. Okay. So the 5 tonsils, okay, there's a left and a right palatine. There's a left and a right lingual. Okay, lingual is for language. Language uses the tongue, so it's behind the tongue. Okay, palatine is the palate, hard palate. So the tonsils are at the back of the hard palate. The pharyngeal is really in the nasal pharynx. Okay, so it's back into the mouth and up into the part of the throat that is exposed to the nasal cavity. Okay, and there's one of those. It's called your adenoids. Your adenoids are up in the airway. So sometimes you have your tonsils, your adenoids removed because they can become inflamed. And if they become inflamed and enlarged, they can potentially provide a restrictive airway, and that can lead to choking or loss of airflow. So this is kind of a half cross section through a person's face. You can see the nasal cavity up above, you have the oral cavity, all the way to the back, you have the pharynx. Think about the pharynx as the elevator shaft that goes from the upstairs to the downstairs to the basement. Okay, the pharynx. And the pharyngeal tonsil is here. You have the palatine tonsil, and at the base of the tongue in the very back is the lingual tonsil. 2 lingual, 2 palatine, and 1 pharyngeal. Okay, and they're positioned conveniently at the place where air and food and drink are entering the body. And so these tissues are continually sampling all those substances that are entering the body. So lymphoids, lymph nodes, thymus, and spleen, we're going to be talking about those. Let's talk about lymph nodes. Okay, this is the overall generic structure of a lymph node. And the first thing you're going to notice is that there are some vessels that lead into the lymph node. We call these the afferent vessels. And then there's one singular vessel that leads out of the lymph node. We call this the efferent vessel. So afferent and efferent, we've heard those words before with regard to the nervous system. Afferent always is coming into the CNS, and the efferent is going out of the CNS. For the same principle, we have the approach and exit. Okay, so if those words help you think about afferent comes first, you're entering, so afferent is approach, okay? Efferent is exit, okay? It's just the flow of lymph in and out of a lymph node. And you'll also notice that there are multiple afferent vessels, that is, there are multiple vessels entering into a lymph node, and then a singular, usually efferent vessel that's going to continue the flow down the pathway on its way towards one of the lymphatic ducts, which would empty into the subclavian vein. So those are the vessels as they're attached. You can see that it has a blood supply, some arteries and veins. They're very small, arterioles and venules. And then the interior here of the lymph node, there's a couple extensions of the outer capsule. There's an outer capsule here made up of connective tissue that protects the lymph node and keeps it encapsulated, because there's a lot of important cells that need to be kept inside, and these can't be disrupted. So the capsule is quite tough, and it helps to retain all those immune cells inside of it. Extending from the outside capsule into it, we have these trabeculae. Trabeculae just means kind of a half wall that is breaking up the lymph node into different portions. This outer region, as in all parts of the body, there's a cortex. Cortex means the outer shell. And then in the very middle is the medulla. Okay, the medulla here, the medullary sinus, is the very middle of the lymph node. And then in this double black dotted line is what we call the deep cortex. So you have outer cortex and then the deep cortex and then the medulla. All right, and then you can see these centered areas, which we call a germinal center. Okay, so here's a germinal center, here's a germinal center. And in these outer part of the cortex, we have collections of B cells, okay? B cells live here. The T cells are living closer to the deep cortex. And so an antigen-presenting cell, like a macrophage or dendritic cell, could arrive into the lymph node and then begin displaying or presenting its antigen to any of these B cells or T cells. And thus, if it gets confirmed that this is indeed a foreign pathogen, these B cells and T cells can become activated. So principally, what an antigen-presenting cell would interact with is a T cell, a T helper cell, which would be the CD4 positive cells. Right? Now, what I want you to think about is, so all of this tissue here in pink with the little black dots, those are all lymphocytes, B cells and T cells. And the analogy I want you to get in your mind is a cluster of grapes that you dip into a stream of water. Okay, now I have a picture of like a faucet like pouring over the grapes in the photo on the slide. However, it's more appropriate to think about the cluster of grapes being dipped into a flowing stream of water. What does that represent? Well, the grapes represent the lymphocytes, okay, B cells and T cells, and they're attached to reticular fibers. Okay, reticular fibers are part of reticular tissue. It's a type of connective tissue, and it's like the stems of the grape cluster. Okay, they hold onto the grape and they kind of keep them together. Okay, so they can kind of move about, but they are organized by the stem. Okay, like the B cells and T cells are organized by the reticular fibers. Okay, clusters of grapes attached to the stem. Now, the flowing stream that's flowing past the cluster of grapes is like the lymph. Okay, the lymph is continuously flowing past the B cells and T cells. And because the B cells and T cells are attached by these stems of reticular fibers, they don't wash away with them. Okay, so the lymph flows past them, and therefore that's exposed to anything that is contained in the lymph. Okay, like any cellular debris or pathogens or microbes or any other white blood cells that could be traveling along in the lymph. And along the way, the B cells and T cells are surveying what is in the lymph. Okay, is there anything questionable? They're always continually checking the contents of the lymph. And as the lymph continues to flow from the afferent through the lymph node into the efferent, the lymph will continue up into the subclavian veins. Here's just another diagram showing you a very similar set of information about the cortex in this kind of olive green, the paracortex or the deep cortex in the light green, and then the medulla is in the middle. Okay, the medulla is where there's macrophages and plasma cells, and the outer cortex here is mostly B cells. Okay, and then the paracortex or the deep cortex is the T cells and dendritic cells. Okay, but for all intents and purposes, if you think about a lymph node is a collection of B cells and T cells, and that's where antigen presentation is occurring. That is, an arriving macrophage or dendritic cell comes displaying some antigen that it's killed, phagocytized, and it's showing and it's communicating a message from the peripheral tissues to the lymph node in an attempt to amp up an immune response, okay, an adaptive immune response. Here's a zoomed in portion on the germinal center. If the B cells get activated, then the B cell population will begin to divide into this what's called clonal expansion. Okay, clonal expansion means that one B cell that got activated will begin to make many copies of itself and then produce a bunch of antibodies. These are just some terms about the parts of the lymph node. Okay, the function of the lymph node is a filter. Okay, so as that lymph is percolating through the lymph node, it is being purified, it's being sampled by all of these B cells, T cells. Okay, removing debris and pathogens. In some cases, there's phagocytosis occurring in the lymph node. And 99% of antigens are captured in the lymph node. So antigen presentation occurs inside lymph nodes, okay? Antigen presentation is what a dendritic cell, here shown in purple, or a macrophage, after phagocytizing and invading pathogen, It can travel to the nearest lymph node and then display or show a T helper cell, okay, a CD4 positive cell. And you can see lots of these pictures of various cells with kind of these cellular or protein projections kind of reaching out and touching each other. Part of that is antigen presentation. Part of that is called co-stimulation, okay? Co-stimulation is a double check to verify the authenticity of the type of message that's being communicated. Okay, so there's multiple proteins kind of reaching out and touching to double check the correctness. Is this indeed the message that I want to send? Like, here is a foreign antigen, or this is self antigen, and there's multiple communications and they all have to match, or there's not going to be a response. Okay, the immune system has to be very careful about what it calls a foreign invader. And so there's what's called co-stimulation. Okay, there's these multiple proteins that are reaching out and communicating between dendritic cells or antigen presenting cells and the T helper cells and other B cells. Okay, so all that to say is the adaptive immune system is very careful about when it becomes activated, and it's based on the co-stimulatory proteins. All righty. So let's move from lymph nodes into the thymus. The thymus is located in the mediastinum, in the superior part of the mediastinum. It atrophies after puberty. Atrophy just means to regress in size, and it plays a smaller role in adulthood because most of the antigens we're exposed to as children. Okay, most of the types of bacteria and types of viruses we get exposed to as a child. And so there's a diminishing role for the thymus into adulthood. Okay, there's kind of two lobes here, the right and the left lobes, and there's some special cells called the thymic epithelial cells in the cortex that encase and surround the cortex. Okay, those cells maintain the blood-thymus barrier, okay, which we talked about in a previous chapter on important barriers between the blood and any tissue. Okay. The thymic hormones are secreted. The thymic hormones are communicating a message about activating and preparing T cells, okay, and stimulating T cell differentiation. That is, taking these T cells to school, okay, initiating the education of T cells. Okay. The thymic Hassel's corpuscles, I'll show you a picture of those. Thymosin is the principal hormone that you need to know about for the thymus. Thymosin promotes the development of lymphocytes, okay, and maturing them into a mature T cell. Here's a picture of the location of the thymus. You can see it's here in the mediastinum. It's in the upper part of the mediastinum, and it overlays the trachea. Trachea is the windpipe, leads from the throat to the lungs, and it lies just above the heart. Now notice just superior to the thymus is the thyroid. Thyroid is not in the thoracic cavity, but is located really in the neck, which is not a cavity. Okay, but there's it also lies over the trachea. Okay, thyroid is superior, thymus is inferior. We're going to do a cross section through it. You can see again a very similar lobular structure as we see in lymph nodes. We're going to see it in the spleen. It's a very similar arrangement of B cells and T cells. Okay. Here's the thymic corpuscle with the thymic epithelial cells that form the corpuscle. Right now, thymus, that's where T cells are differentiated and developed. Okay, that's really all you have to know about the thymus, is that it's involved in the maturation of T cells. Spleen, okay, the spleen has three functions. We've already talked about the first function, which is the removal of abnormal blood cells, essentially, especially the red blood cells. Although it is also white blood cells, it's mostly the red blood cells. Number two is the storage of iron. Remember, when we are processing red blood cells, we're lysing them, and the hemoglobin gets broken down into bilirubin. The iron center of hemoglobin gets recycled and gets brought back to the spleen, and then eventually that gets processed and brought back to the bone marrow so we can make more hemoglobin. Third function here is the initiation of an immune response by B and T cells. Okay, so three major functions here of the spleen. Okay, red blood cell graveyard, processing iron, and then three is the third function here. I want you to think about, it's kind of like a big lymph node. Okay, the spleen functions like a large lymph node in that you have antigen presentation occurring, that is a dendritic or a macrophage doing antigen presentation and then activating B cells and T cells. Okay, but the fluid that a spleen processes is not lymph. The fluid that a spleen processes is blood. Okay, but it functions in the very similar fashion. Right? It's held in place by the gastrosplenic ligament. It's how it's held there, and there's blood supply that's going in and out. So this is the location. It is just found on the left side. If you imagine that the pancreas is like a gun and it's pointing at the spleen. Okay, so here's the pancreas. It lies right in the crook, this kind of curvature of the duodenum, which is the initial segment of the small intestine, just distal to the stomach. The stomach here has been resected, dissected out. Okay, and here's the pancreas. Pancreas points to the spleen. The spleen lies on the left posterior border of the abdominal cavity. Okay, it's below the diaphragm, makes contact with the diaphragm, with the stomach, but is located on the left side. This is a cross-sectional view, transverse view through the abdomen. You can see the stomach, the liver, the kidneys, there's the pancreas, and here on the left side is the spleen. Now, this is probably a confusing picture if you're thinking this is a superior view, but it is not a superior view of this transverse section. It is an inferior view. And the illustration here kind of hints at that, because the way that this little blue glass plate is kind of slicing through the body gives the sense that we are looking at the underside of that. So this is an inferior view. That's the only way that this makes sense, that the liver is on this side and the stomach and then the spleen. Okay. Here's another look at the inferior view of the spleen. This is called the hilum. The hilum is where all the important tubes are entering into the spleen, like the lymphatic vessels and the blood vessels. Okay, let's keep going. Inside the spleen, histologically, we have two types of tissue, the red pulp and the white pulp. And as you might imagine, the red pulp is where we're processing red blood cells. Okay, that's the red blood cell graveyard, as we talked about. The white pulp resembles lymphoid nodules, like a lymph node or the tonsils. Okay, so the white pulp is where you're doing antigen presentation and lymphatic function. The red pulp is where we're doing kind of blood cell processing. Okay, so this is a picture. It's hard to distinguish from a histological section. I will not ask you to do that. Okay, here's a diagram. Again, diagrammatically, you can see the difference between the red and the white. And functionally, it allows for a sorting of those functions into process red blood cell death and turnover versus doing B cell and T cell activation. All right, so that was a review of some of those lymphoid tissues. Now let's start going into immunity. Okay, what is the big picture of immunity? Okay, it's the body's ability to resist infection and disease. Okay, the body's ability to resist infection of a foreign pathogen, microbe, and the disease that could result from infection. Two parts here: innate immunity and adaptive immunity. You might hear them referred to innate as nonspecific and adaptive as specific. Okay. So these are the aspects here of innate. They always work the same way. It's against any type of invading agent. It's a very rapid response, and it's a non-specific resistance. Okay, you don't need any special tools to do this. All of our phagocytic cells can do the job. The adaptive immunity protects against specific pathogens, depends on activity of lymphocytes. You need B cells and T cells for adaptive. And it's a delayed response, okay, on the order of a couple of weeks. It takes a couple of weeks for this to get activated appropriately, because the B cells and T cells that activation takes some time. Okay, and it provides a specific resistance. So although it takes some time, it is much more rapid response to a second exposure. So the first exposure, it'll take some time, but on the second exposure, third, fourth, fifth, any subsequent exposure, the adaptive immunity, it's time to shine because it's already developed the sensing apparatus and the tools to deal with that infection, and it responds very rapidly after second, third, or fourth exposure. All right, so first we're going to talk about the non-specific defenses, okay, part of the innate immune system, okay, the innate immune system. And part of this, as I said before, is the physical barriers, our skin and our mucous membranes, okay? Our skin and our mucous membranes, all the fluid parts that are protecting us from the outside. Our tears do some of that, our snot, our mucus in our mouth and our nose, all of those are physical barriers that are intended to stop or trap pathogens before they get inside of our body tissues. We're also going to talk about phagocytes like neutrophils, monocytes, dendritic cells. They perform phagocytosis. We'll talk about immune surveillance due to natural killer cells. Interferon and complement are two protein tools that are used during innate immunity. Okay, interferons are a set of proteins, and complement proteins are a set of proteins that can participate in innate immunity. The inflammatory response, we'll talk about a typical inflammatory response, the 4 cardinal symptoms of inflammation: rubor, calor, dolor, tumor, and then we'll talk about fever. Okay, as a last resort, if the body is still not managing this infection very well, then we can go full bore, full system fever, like the body gets hot, and that increased temperature is an attempt to speed up the immune system and really inhibit the growth of the pathogens. All right, so here is a little bit more illustrated detail on these various aspects of innate immunity. Okay, our barriers we're going to talk about. The skin, keratin layers are important. Sweat has certain antimicrobial properties: sweat, tears, mucus, earwax. Phagocytes, we're going to talk about the various cells that can perform phagocytosis. Phagocytosis is the chief way in which foreign microbes get dealt with, okay? To be stopped, you're going to engulf these foreign invaders using one of these phagocytes. You have fixed macrophages versus the migratory macrophages like monocytes. Eosinophils can undergo phagocytosis. Fixed versus free. Fixed just means it's a resident macrophage. It's usually in the tissue where it's found and it just stays there, okay? It doesn't move. The free macrophage means it can move in and out of the bloodstream, okay, or in and out of the lymphatic system. Neutrophils are, of course, the most abundant white blood cell, and it performs phagocytosis and is a first responder to bacterial infection. Immune surveillance, the natural killer cells are all the time continually surveying your body tissues for appropriate documentation, whether your cells are expressing your MHC class 1 receptors showing self-antigen to double check, do you have your papers? Do you belong in this body? Otherwise, I'm going to attack you. Interferon, okay, interferon is a protein tool. As I said, it's something that the cells are secrete in order to fight viral infection. Okay, that's the thing that you need to know about interferon. It is a protein that's secreted to fight viral infection. We're going to have a review of all the white blood cells, including some mast cells. Mast cells are located in the integumentary system. Okay, they're in lots of peripheral tissues, but most notably in the integumentary system. They help with signaling an inflammatory response. Natural killer cells, basophils, monocyte neutrophils, your B and T cells, those are your lymphocytes, macrophages, and eosinophils. We've seen those before. The only other one that we haven't talked about was the dendritic cell. Okay, the dendritic cell is very similar to a macrophage. Okay, it doesn't look like it, but the dendritic cell can perform phagocytosis and is typically a professional antigen-presenting cell. Okay, it's a professional, meaning that's the thing that it does. Along with macrophages, it engulfs, breaks it apart, and then travels to a lymph node or other lymphoid tissue and communicates the message about a foreign invader to the adaptive immune system, specifically T helper cells. All right, so physical barriers, phagocytes, immune surveillance, we've talked about these, okay? Continuing in this illustration here, complement, okay? Like interferon, it's a protein, it's a secreted protein that can become activated and help with mitigating a pathogenic infiltration. So the complement cascade can activate these proteins, and there's a sequence of events whereby they get activated and they can form something called a MAC, which is the membrane-associated complex. Okay, membrane-associated complex is essentially like a big drill. It bores a hole into the Mr. E. coli here, causing the cell to lyse. The inflammatory response, we'll talk about how mast cells play a crucial role in triggering or stimulating an inflammatory response in the tissues. Lots of things here to do with blood flow and increasing the permeability of those vessels to increase blood flow to the site, increase lymphatic drainage, recruit more phagocytes, and in some cases participate in the fever response, okay? Fever is really a systemic response to infection. After some time and the immune response is not able to mitigate the pathogens, then we can tell the brain, hey, we're struggling with this infection. Let's go full systemic, and the whole body will begin to heat up, raise the body temperature in an attempt to speed up the immune response, as well as mitigate or slow down the growth of microbes. Interferons and complement, we'll talk about those. So this is the steps here in an inflammatory response. Okay, you can see the numbers 1, 2, 3, 4, 5, 6, 7. Okay, so the first part of an inflammatory response here is that there's some compromise typically in the skin. Okay, we're talking about an infection here in the integumentary system. There's been a scratch, a scrape, a puncture, a cut, and bacteria are now on the premises, as it were, and infiltrating the tissue. The mast cells and the resident macrophages can detect the presence of these foreign pathogens and become activated. Okay, so the clotting cascade will begin to get activated to stop blood flow if there's an injury that's causing bleeding. So that's going on, and at the same time, the macrophages are in the mast cells are communicating using cytokines to the nearest blood vessel or capillary to recruit more phagocytes. Okay, so it's sending a chemical messenger. Mast cells secrete factors that mediate vasodilation and vascular constriction. Okay, so vasodilation would cause increased blood flow, and that increased blood flow, ironically, because you're thinking about platelets and you're going to cause decreased blood flow, but stopping bleeding is the early stage. And after that early stage, there's an inflammatory response. So the clotting happens very first thing, stop the bleeding. And then after the clot is formed, then we can increase the blood flow. It gets warm, gets red, gets sore, gets swollen. And that will increase the blood flow after bleeding is stopped to facilitate and encourage additional immune cells arriving to the site of injury to participate in phagocytosis. Okay, the neutrophils are the main group of soldiers that are fighting that battle. They show up, they phagocytize, and then they can do oxidative burst. You can do phagocytosis. There's lots of ways that neutrophils can do that to stop the growth of the pathogens. Okay, what you're really, the race here is a time. Okay, there's a race against time, because as soon as those pathogens, let's say it's a set of bacteria, they enter the body, they're going to begin feeding on the nutrients that the body provides. And in that safe environment of a warm, moist environment, they begin to undergo cell division. So the microbes want to eat, grow, and divide. Okay, eat, grow, and divide, and soon, and you started with 100 cells, and now you have 1,000 or 1,000,000 cells, because they're eating, feeding, and dividing. And so the immune response has to be fast to stop the microbes' cell division, okay? Because bacteria like to divide and grow very rapidly. So it's a race against time to stop that cell division. Okay, so number 4 here is the neutrophils secrete factors that kill and degrade the pathogens. Number 5, neutrophils and macrophages remove pathogens by phagocytosis. And then macrophages, you have resident macrophages that are playing a role. There's recruited migratory macrophages that can then engulf, phagocytize, and then bring the message of this infection to a lymphoid tissue. Okay, so number 6, macrophages recruit immune cells, and 7, initiate tissue repair. So this is all part of the innate immune system. Okay, where the macrophage then plays a role as part of the adaptive immune system, it's going to carry the information from its phagocytized microbes, carry that information to a local lymph node or the spleen, and do antigen presentation and continue on with the activation of the adaptive immune system. All right, so here's a whole table looking at all of the steps that go on during inflammation. And these 3 red boxes here indicate the 4 cardinal signs, okay? 4 cardinal signs of inflammation: heat, redness, pain, and swelling. And there's 4 Latin words that have a similar sound. Heat is calor, okay? Heat is like calorie. Redness is rubor, red. Pain is dolor, and swelling is tumor. Okay, tumor just means a swelling. Now we need to think of a tumor as like cancerous, but it just means a swelling. Okay, in this case, swelling of fluid. Okay, so calor, rubor, dolor, and tumor. Those are the 4 cardinal signs of inflammation. Increased blood flow, heat. Okay, all of those are produced by increased blood flow to the tissue. It's a localized response. Okay, if you have a little cut, just the area around it will get red and swollen and painful. The pain comes from the increased blood flow. The fluid that's entering the tissue is pressing on the pain receptors. So it's going to be tender, okay, the pain response. If the inflammation increases, you'll get the 5th sign, which is not the cardinal sign, but the 5th sign would be loss of function, or even joint, limiting the joint function. Okay, joint movement can be impaired. So if you have lots of trauma to a body part where there's a joint nearby, you can have decreased mobility of joints because it starts to affect that as well. All right, this is a picture of a macrophage. Okay, it could be a macrophage or a dendritic cell. Okay, both participate in phagocytosis. Okay, the green pathogen here has some PAMP shown in yellow. Okay, this is a pathogen-associated molecular pattern, and it recognizes that non-specific pattern like LPS, lipopolysaccharide. It engulfs the pathogen, and then it fuses with what's called a phagosome. The phagosome is a cytoplasmic vacuole, okay, that contains digestive enzymes and other reactive oxygen species that can obliterate, tear apart, and destroy the engulfed pathogen. After which, a small part of that torn apart pathogen gets associated with the MHC class 2 receptor here shown in blue. Okay, MHC receptors are super important. Okay, there's class 1, there's class 2. We're going to talk about them both. And in this case, because we're talking about a macrophage, it's going to be the MHC class 2. And this is, you can think about the MHC class 2 as the silver platter on which, you know, the cell is like, hey, look at what I just captured. Okay, my marker represents a little bit of the E. coli. Okay, maybe it's a little part of the cell membrane, and it displays it on its surface like, hey, I just killed this bad guy, look at it. And it can travel and show this pathogen to a T helper cell. That's called antigen presentation. MHC2 always displays foreign antigen. It's showing the T helper cell, this is a foreign invader, this is what it looks like. So let's respond to this bad guy, and here's the molecular profile of what it looks like. Okay, here's a dendritic cell showing a lymphocyte is doing antigen presentation here. All right, so phagocytosis, there's 2 really big outcomes here. 1, the microbes are killed because when you phagocytize them, you're kind of engulfing them and you're tearing them apart. Okay, stop them now. The other major part of phagocytosis, and this only applies to the antigen presentation cells, is that the digested pieces of the microbes are subsequently presented to the innate immune system. Okay, so instead of stop them now, it's stop them again later after a second or a third exposure to that pathogen. Okay, stop them now, stop them later. Fight the battle now, fight the battle later. All right, so PAMPs and DAMPs. Okay, so this is associated with the innate immune system. The PAMPs and DAMPs are it's something on the surface of a microbe, okay, a pathogen that is recognized by a phagocyte. Okay, our chief phagocytes, remember, are neutrophils and our monocytes and dendritic cells. Okay, so a PAMP is a pathogen-associated molecular pattern. It's just something that a phagocyte can recognize as foreign. Okay, PAMP. A DAMP is a damage-associated molecular pattern. It is similar, has a similar role, but it's not from a foreign cell, it's from a self cell. Okay, so if your cells are in trouble and they're lysing, you're undergoing necrosis, you have a necrotic cell death, your cells are dying and breaking open, these DAMPs will be released. So this is called a damage-associated molecular pattern. Okay, so it's again, it's a warning signal like, hey, immune system, my cells are dying and breaking open, and these DAMPs are kind of exposed, and then your immune cells can respond to them. So PAMPs and DAMPs are signals, okay, that communicate to the immune cell, let's get activated. Okay. A PRR, pattern recognition receptor, is a toll-like receptor, TLR. Okay, there's also C-type lectin receptors and NOD-like receptors. All of these have a very similar role. The toll-like receptors are the ones you probably hear about the most. Toll-like receptors can identify a PAMP or a DAMP. They bind to a PAMP or they bind to a DAMP, and then once they bind, they get activated. Okay. So here you can think about innate immunity as participating in response to strangers, okay, foreign microbes entering, versus dangers. That is your own cells that are stressed or damaged or going through necrosis. Okay. So the PAMPs would be some molecular pattern associated with a pathogen. The DAMP would be some intracellular protein or marker that only gets released when there's cell damage. Okay, so both the PAMPs and the DAMPs can stimulate the immune system to respond. All right, so here's examples of some PAMPs. Okay, so you have bacteria that can be gram-positive bacteria or gram-negative bacteria. Okay, LPS is a big one. Okay, the lipopolysaccharide of gram-negative bacterium. Flagellin is a big one because the only cells in the body that have a flagellum are sperm cells. And in fact, that's an important side note that we'll talk about in the reproductive system. Lipoproteins, peptidoglycans, lipoteichoic acid, these are all examples of PAMPs. Virus proteins that are on the surface, parasite has GPI anchors, yeast cell have different protein markers on their surface. All of these are just examples of a PAMP, okay, to which a macrophage or a dendritic cell can say, hey, I recognize that. That's not supposed to be here. Let me go eat you. And therefore, when it sees the PAMP or sees the DAMP, it's going to start communicating an inflammatory response and trigger an antigen presentation pathway. Okay, macrophages are big eaters. You can have fixed or free. Okay, fixed macrophages are often called histiocytes. Okay, they stay in the tissue. Free macrophages are roving around. They can wander throughout the body. Okay, that's the only difference there. Something about resident macrophages. This will be the last point because I'm out of time. Okay, resident macrophages, they live in a specific tissue. Okay, they're a macrophage. So you have a macrophage that lives in the brain. We call those the microglia. A macrophage that lives in the liver, we call it a Kupffer cell. Macrophage that lives in the lung, we call it a dust cell or an alveolar macrophage. Okay. There's various other, in the Langerhans cells are in the skin. We have other cells for the kidney, the peritoneum. So all these tissues have their own specialized macrophages that they develop in the bone marrow. They travel to this tissue of preference, and then they just start to specialize for liver-associated pathogens or skin-associated pathogens or lung-associated pathogens. These are the ones that you need to know: microglia, Kupffer cells, alveolar macrophages, also called dust cells, okay? Because it makes sense if you breathe in dust, then the dust cells get activated. Kind of a play on words there. And then the Langerhans cells are found in the epidermis, okay? They're all examples of a resident macrophage. All right, I'm going to stop there because I'm out of time. See you guys in class. End this thing.